And I want to thank George and Angelina Kostas for establishing the Research Center and the family for the continuation of it. And I hope in this brief presentation to give an overview or to illustrate what this sort of funding can bring to us. So I'm going to talk about mitochondria and transplantation of mitochondria. Unlike a heart, liver, kidney, or whole organ transplant, or even a cell transplant such as islets, which we've been involved with, this is an organelle, a piece of the cell going in to try to re-energize cells. And it's a project, and we seek NIH funding or other competitive funding. They say, well, we need pilot data. Hence the importance of the Kostas funding and what it has meant to us. And I hope this brief presentation will help illustrate that. The title of this is Re-energizing the Failing Heart. And in terms of disclosures, I have none competing or conflicting disclosures to declare. And we're going to talk about energy and biologic energy. Very similar to external energy that drives our lives, that runs warms our houses or cools our houses. If we're in Houston, warms them in Boston, I guess. That fuels our automobiles. I mean, it's no different. Biologic energy is essential, and the delivery and processing of it is very similar to what we see with the external energy. It needs to be processed, harvested out of a farmer's field. Then it needs to be put into a form that we can utilize. Whole wheat bread or a McDonald's hamburger. And then taken in, processed, transported to the cells, and then within the cell, then converted into something that can be utilized for that cell. And those cells that you show that were heart cells that were contracting, or the other muscle cells that were moving, require energy. It's not going to happen without energy in the form of ATP. So biologic energy, of course, has its effect in our health in three major categories. And of course, the first one is one that we're very acutely aware of, and has been an epidemic of the storage of energy. The other two, though, fueling neoplastic or cancer or even non-cancerous tumor growth, requires a lot of energy. And that energy cannot be created. I mean, these tumors cannot violate the laws of thermodynamics, the second law in particular. It has to extract energy from normal cells and pull it in to its own devious use of cell cycle growth, dividing, growing. And at the same time, your well-differentiated cells or muscle cells, brain, are losing energy. So it's being transferred. So there's a fuel problem and energy there. And that's one of the areas we're looking at in the lab. It's a bioenergetics lab. But what I want to talk about today is a high energy organ that's out of energy or losing energy. And the two highest energy using organs, of course, are the brain and the heart. And a major project in our lab, and one that the Kostas has been helping to fund, is to try to re-energize failing heart in a rather unique way. Heart failure is relevant because it's quite prevalent. And if we look at individuals over the age of, if we focus on Houston alone, and we look at individuals over the age of 65, the elderly, Henry, I'm in that club. The incidence of it is 10 in 1,000. So just in the Houston area, there are 20,000 new cases of heart failure in individuals over the age of 65. And what a lot of people don't realize, and it doesn't get the attention of cancers, but it has the same 5-year mortality rate as breast cancer. 50% of those individuals will pass away, in spite of everything that we have to offer them today, will pass away because of the heart failure within 5 years. And this has been well documented, not just in Houston. It's a national health problem. the significance of it and the rationale for the work we're doing as a bioenergetic lab is to try to re-energize it. Stephen Neubauer from Oxford published this review in 2007 that calls it the failing heart engine out of fuel. Came under a lot of criticism because it's not a fuel problem, at least as we might think about it. That is, most individuals with heart failure have hyperglycemia, so there's a lot of fuel circulating in the blood. The fuel is not being processed. And he got a lot of criticism for this, and then he came to say, well, by fuel I meant the ATP that goes from the mitochondria to the muscle so that it can contract. But clearly he has shown, and this 2007 review was a result of work that he began in 10 years earlier, in 1997, as a publication using MR spec P31 to show that the failing heart is out of energy, out of ATP, the compound that provides the energy for that muscle to contract. And if we look here on the lower bottom as a volunteer, the PC ATP ratio greater than 1.6. And I don't want to dwell on that too much, other than to say that what is shown here in the waveform that is the highest, the PCR, is ATP, the energy of that phosphate being transferred out of the mitochondria into the muscle of the heart. And it needs that reservoir, because once it goes to ATP for the contraction, which is the third peak here, which is the third peak here, it has a very short half-life. It doesn't last long in the cell, so you need that reservoir. And when you see the ratio of those two changing, there's a poor outcomes. And as we go from 4, 3, 2, 1, the top one there, you can see you're losing that relationship, still making the ATP up here, but losing the PCR. There's no reserve. And that individual passed away 7 days after this study was done. And I've talked to Neubauer about this, and he had started this work in the 1990s, published this in 97, has continued it. And then in his review that came out in 2007, he had, as part of it, this chart here that shows that indeed ATP, which is and the storage of ATP, which is the phosphocreatine, decline. This one's declining faster than that. So over time, they are declining. And the substrate, or the fuel itself, the oxidation of it, or the burning of it, the taking of the electrons from the carbon bonds in glucose or in fatty acids, which happens in the mitochondria, is declining, even though the supply is there. You measure triglycerides, you look at fatty acid levels, which we've done and published in the heart. The fuel is there. It's just an upstream problem. It's not getting processed. And looking further, and this is when we started our studies about a year or two after this, and I talked to him about this, that in here there's respiratory chain complexes, ATP synthase, oxygen consumption, uncoupling proteins. These are all mitochondria-related things, and you can see that they are shrinking in time. So we asked ourselves, are the mitochondria permanently damaged in heart failure? And this, again, just more information from his paper. Are the mitochondria damaged in heart failure? So we started out in 2008, 2009, trying to isolate mitochondria from hearts. Small animals were relatively easy, it's been done. But human hearts, where you need 500 milligrams to a gram, something larger than an olive, you can't do it on a simple biopsy. Or we can now, we've developed techniques, but we couldn't then. You can't just do it unless the heart is coming out with the sample. So, and then the mitochondria need to be isolated within 30 minutes to an hour, so you need geographic location. we're 5 floors up from the operating room. So this is just a stain to show that there's a nucleus here, but these cells are packed with mitochondria, stained red here with JC1. It has a positive charge, it's a cation, it's attracted to the negative matrix of the mitochondria. So they're plentiful, and these are normal cells here. And we can isolate the mitochondria, and this is just an example of a normal cell, how we would study it using a high-resolution respirometry, it's called. And mitochondria will take oxygen, and which is the rate of oxygen consumption. You're converting oxygen, reducing it to water, is reflected here. If the mitochondria are working, then you're going to get curves like this based on as you add substrate in and oxygen. If they were damaged, it would just be flat. There would be no activity. Or if it was uncoupled, it would be just continually burning oxygen. There wouldn't be this up and down. So this is a pattern in a normal cell. So we were able to establish techniques for isolating mitochondria from muscle cells, skeletal muscle, heart, and we'd look at tumors too. And this is an example of a sample, and we've done 50 or 60 of them, then we published them 3 or 4 years ago. First one's to really isolate mitochondria from failing human hearts. And this is, LVAD was put in on this particular patient. You can see the sample we got. We get it from the operating room, a surgeon drop it in here. We carry this up to the lab and immediately start processing it and isolate it. So the geography of it is essential if you're going to look at respiratory function. Of course, we could snap freeze it and look at it, which we do, look at enzymes and genes, so forth, months later. But to do the respiratory studies requires that timing. And early on, we got results like this. These are mitochondrial isolates from, on the top, non-failing left ventricle, and on the bottom, a heart failure ventricle. And we were shocked because we expected either to see it not functioning flat or that there'd be a lot of excess oxygen consumption, but no coupling to it. Or by coupling, I mean up and down. And we didn't believe it. So we looked at our procedures. After 5, 10 of them, presented it at various seminars and societies, and finally we've come to believe it, and we've published it, and it took almost a year to convince the reviewers of our techniques were solid, and you know, but we did get it published in the Cardiology Journal, showing that there are functional mitochondria still in advanced chronic heart failure. But obviously, they're not supplying the energy as I showed earlier. And we looked at gene expression and mitochondrial function. This kind of boring it. This is from one of our papers. I don't want to get into the details of it. Filter keys, probably not. Huh? Okay. It may be a little hard to see, but we looked at the same heart. These were paired samples that come in, advanced heart failure for survival, an LVAD, mechanical assist device was put into that ventricle. A little plug of the tissue came up to the lab. And then the patient was kept alive for, on average, 6 or 7 months. We had 9 of them here. And then they would undergo, there's a bridge to transplant, they'd undergo transplant. So when that heart with the LVAD came out, we got tissue from that same heart and restudied it and did paired samples and showed not only were they functional, but there was a reduced number of mitochondrial proteins, but they recovered and came back. So there is hope. They are energy deprived, but it's not but there may be some hope there in terms of getting energy back into the cell. So then, on reflecting on this 2 or 3 years ago in a variety of conversations with science friends and stuff, as you say, it's always nice to have a phone call and be able to talk to somebody and get into a project. And why don't we take mitochondria and just put them into cells and transplant them? Of course, when the mitochondria is outside the cell, they're kind of a bacterial lineage and don't survive very well on their own outside the cytoplasm. So we needed some work with the engineers, nano people, to coat them, keep them alive. And we started using a higher throughput machine. This is called Seahorse. And looking at some normal established tumor cell lines, breast cancer cell lines, and so forth. Human cardiomyocytes, we just don't have a technique for growing them in cell culture. In this case, just to illustrate what we did, we took some breast cancer cells and isolated, these are human breast cancer cell line established, isolated mitochondria from a mouse liver, which you think it would reject it right up front, and over time it might. And we just looked to see, did it improve the respiratory function? And on the seahorse, we get a graph like this, but a little closer look at it. The normal cell line is here in the brown. And you can see when we added coated mitochondria, we did it nano-coated, uncoated, different concentration. We're still working on the polymer and what's best to coat them, that it didn't kill them or damage them. There's no straight line. It's not uncoupled. I mean, you can see there's very nice oxidative phosphorylation going on here in these human breast cancer cells getting mice liver mitochondria. And our ultimate goal, of course, and then there's a so-called Warburg effect, where cancer cells have the strategy of taking energy from other cells and metabolizing glucose and bypassing the mitochondria and making lactate. And this measures the lactate production. You can see from the normal cells are very active that way, but when we coat them and transplant it, we drop that and shut off that Warburg effect. And it's illustrated here on this graph here, where we're looking at oxygen consumption rate here. These are the breast cancer cells doing what cancer cells do. They are making, this is the acidification rate. So they're making a lot of acid here and not consuming much oxygen. This is their survival tactic, how a breast cancer or any tumor grows. It bypasses the need for oxygen as they're growing so quickly they don't have the blood flow into them, and they use glucose. They're fermenting, essentially. And a shift this way is very positive. Something that's completely aerobic, like a normal heart cell or muscle cell, would be up in here. And this was a tremendous shift in these cells. This was transplanted with the coated mitochondria. So this was kind of a pilot study and showed the feasibility of maybe we can change the phenotype or the metabolism of cells that have energy, altered energy strategies different than a differentiated cell. And even though it was a xenograft, essentially. So, and this just, it goes on, just looks at it in more detail. Where we had shifted from this area to this area. We looked at several different cell types here, and most of them all neoplastic cells. And we ended that with funding from the Kostas grant and endowment, and it has helped. And now we want to move to the problem of the failing heart. So we've coming back to the heart, which the failing heart, it is out of energy. We have a mouse model that we published earlier this year of a non-ischemic heart failure model. And it's published in a physiology journal, where we can take a 12-week-old mouse and put it into heart failure in about 5 weeks. It's not ischemia. We're not affecting the oxygen flow into it. And this is a model now that we can take the heart, isolate the cardiomyocytes, and then we've actually already started the process. So take skeletal muscle from that same animal and do an auto transplant rather than a xenotransplant, and isolate the mitochondria from this mouse's muscle and see if we can put it into that failed myocardium and improve. We have a look at contractility, very similar to what you had showed there, and we can quantitate that. So that's kind of an example of where we're going with it, and is trying to see if we can't take conditions like heart failure and go beyond our current therapy, which just has reached its limit. There's not, in the past 5 or 10 years, been very little advances in medicines. The mechanical devices are getting more efficient and are getting smaller, but ultimately that's not the answer. And something with a mortality rate that high. So funding like this has given us an opportunity to look at energy transfer in cells, and failing heart is one example. But we're also, when you look at this mechanistically, though, it may give us opportunities of. You can imagine if somebody had a breast cancer, for example, and came in, you could, instead of giving chemotherapy, take some of that of her mitochondria, a small sample of it from muscle, and infuse it into the tumor and change the characteristic of that tumor. And we've looked at growth and aggressiveness of these tumors, and it completely changes them. Yeah, it's a little bit on the lunatic fringe in a way, but in the other way, if you think of personalized therapy, it's not. And this funding has really helped us move this way, and we're hoping to, I think we're getting enough data now to look at some NIH or other external funding on it. We want to understand, mechanism, energy capture, how it gets in there, transfer, supply. I mean, it's, and this would apply, another major area is the brain, and a lot of degenerative brain conditions, including Lewy body disease or Alzheimer's, Parkinson's, have mitochondrial disorders. So we have a team in the lab. The lab has grown, as I move more from the clinic into the lab, starting 10 years or so ago, we've grown to where we have 5 scientists, and each one of them having teams of 2 or 3. So we have 22 in our Center for Bioenergetics. A lot of my colleagues aren't quite sure, is this an anti-aging clinic or is it a research lab? But it is a research lab. We've got some very nice scientists. Anisha was involved from the beginning, PhD in physiology, but she studies mitochondria and muscle metabolism. Ajun Zhang, Shuman Lee. We've collaborated with Alvin Blanco and the Nanomedicine team, Zhuhong. And of course, we'd never happen without the people I can pick up the phone and call, the cardiovascular surgeons. And now with robotic stuff, they're more available to talk on the phone. They just hang out. And they love these projects. And of course, the cardiac heart failure team has been very central and been part of this. So